Hello everyone. This video is for a B.Sc program 6th semester chemistry paper 8. In that metabolism chapter, this session is dedicated to protein metabolism, session number 35. See, in this you are going to learn about what are proteins and amino acids and in detail about the amino acid metabolism. See, all of us know very well that most of the nitrogen in our diet is derived from proteins. And these proteins are large molecules and hence cannot be absorbed by the small intestine. So then how does the metabolism of protein takes place? They are hydrolyzed to their constituent amino acids with a combined action of proteolytic enzymes. Okay. So remember, digestion of protein occurs in three organs, that is stomach, pancreas and in the small intestine. So we'll begin to study the metabolism of proteins now. Before understanding the metabolism of protein, we will see now what are proteins. So as I told just now, proteins are the macrobiomolecules and they are the polymers of amino acids. So you have learned right from your high school chemistry, large number of amino acids combined together to give you proteins. Okay. So a linear chain of amino acid residue is known as a polypeptide. Okay. So a protein contains at least one long polypeptide. Okay. And proteins are long chain amino acids where the number of amino acid residues are more than 20. Now, let me explain you the chemistry of these amino acids. See, amino acids, they are the bifunctional compounds having both an amino group and a carboxylic acid group. So this is C in H2, H, this is COOH and this is R. So R is the side chain. Depending upon the nature of R, okay, which is very specific, we have 20 amino acids. So I want all of you to learn to write the structure of amino acids because to write the reactions, you should know to write which is the amino acid you are taking. So how do you remember this? At the center you write carbon. I told you they contain both amino group and a carboxylic acid group. So write NH2COOH and write one R here. R is a side chain. Okay. And you have to satisfy the valency of carbon, put H here. Okay. So if you know to write this and if you can remember for what R, which is the amino acid, you can easily write the structure of 20 amino acids. Okay. So carbon at the center, NH2 group, COOH group, R here and H. So try to know what is the side chain for the individual amino acids. Okay. So this amino acid metabolism takes place in three pathways. So you should know for each of these pathway along with the example. That is amino acid decarboxylation, amino acid deamination. In that you have oxidative deamination, non-oxidative deamination and transamination. Okay. So we learn one by one what are these transamination, decarboxylation and then deamination. See, we get this nitrogen in proteins from so many nitrogenous substances. So the main purpose of this protein metabolism is this nitrogen, which is in the form of NH2 group, has to be eliminated because lot of this cannot be stored in the blood. So elimination of this amino group takes place through many ways. So what is actually you are doing is you are trying to eliminate an NH2 group from an amino acid, right? Now, see, amino acid metabolism start with the process of transamination. So you should understand what is a transamination. See, as the name itself tells you, transamination means there is a transfer of an amino group. So transamination is the transfer of an amino group between an amino acid and a keto acid is known as transamination. I repeat here, transfer of an amino group from an amino acid. See, this is amino acid. I just explained you how to remember the structure of amino acid. What I have taken here is a zwitterion form of an amino acid. In all the reactions, amino acids enter in its zwitterion form when it undergoes the reactions. So, transfer of an amino group from one amino acid to a keto acid with the help of an enzyme transaminase which requires pyridoxal phosphate PLP. What is formed? A new keto acid. See, in place of NH3 group, you have a CO group here. So you have a new keto acid and a new amino acid. So this process is known as transamination. So to put it briefly, transfer of an amino group from one amino acid to a keto acid which is catalyzed by transaminase in presence of pyridoxal phosphate, because this pyridoxal phosphate accepts this ammonia group, okay, amine group, to form a new keto acid and a new amino acid is known as transamination. So look at this example now. So what is transamination? Transfer of an amino group from one amino acid to a keto acid in the presence of transaminase to form a new keto acid and a new amino acid is known as transamination. Why I am stressing so much is you get for 2 marks question. What is transamination? Explain with an example. Okay. Now, coming to particular example. See, the purpose of this transamination is to convert the amino group present in this amino acid to a glutamate. You see in the examples now. And this glutamate can later undergo oxidative deamination or a deamination process so that this ammonia gets eliminated, which enters into urea cycle. So this is the way how an ammonia or a nitrogenous substance present in an amino acid are finally eliminated through excretion, that is in the form of nitrogen. Now, see, when this is alanine, if R is equal to H alanine, R is equal to H, it is alanine. See, alanine, when undergoes transamination, in presence of alanine transaminase, see, transfer of this amino group to a keto group here. Look here. In place of keto group, you have got an amino group. In place of amino group, you have got a keto group. So, what are the products you have got? You have got pyruvate and then glutamate. You know, pyruvate can later enter into Krebs cycle by converting into acetyl-CoA. Glutamate can transfer its amino group through deamination process. Okay, so we have got lot of glutamate channels in the body. So the purpose of transamination is to convert or to transfer this amino group completely to get glutamate, which in turn can eliminate this NH3 by a process called deamination. The intermediates obtained can either will enter into a TCA cycle or Krebs cycle. They are the intermediates for the Krebs cycle or TCA cycle. Coming to the next example. See, this is aspartate. This is aspartate, alpha ketoglutarate. When it undergoes transamination reaction, transfer this amino group to here. What you got is a glutamate and you have got oxaloacetate which can enter into TCA cycle. So remember these two examples are very important. That is, the first one alanine undergoing transamination, second one aspartate undergoing transamination. So you should know here definition of transamination and examples for transamination reactions. Now coming to the next one. See, we have got amino acid here. The process starts with transaminase reaction. So you got glutamate. Glutamate can eliminate ammonia, which can enter into urea cycle, where finally this nitrogen is excreted. So this gives the whole picture of protein metabolism. This entire slide will give you the whole picture of protein metabolism. Okay. Now, coming to the next process, decarboxylation. You know very well decarboxylation means elimination of CO2. Elimination of CO2 is known as decarboxylation. In this amino acid, you have the COOH group which has to be eliminated. So after this eliminated by an enzyme amino acid decarboxylate, what you get are the amines. The importance of this decarboxylation is whatever the amine we are going to get here are physiologically active amines, and many of them are the neurotransmitters. So you should know the examples for decarboxylation. So amino acids undergo decarboxylation in the presence of enzyme amino acid decarboxylase to form primary amines. Now, coming to a particular example, see when serine undergoes decarboxylation in the presence of serine dehydratase, in the presence of pyridoxal phosphate, you got ethanolamine. Similarly, histidine, which is an amino acid, on decarboxylation produces histamine, which is a powerful vasodilator. Similarly, 3,4-dihydroxyphenylalanine on decarboxylation produces dopamine. Decarboxylation of tryptophan produces serotonin. See, this dopamine and serotonin are called as happy chemicals. They have got multiple physiological action, and for any human being to be active and to be a normal person, we require both of these compounds. Dopamine and serotonin level should be properly maintained in the body. Okay, they have got multiple physiological role like pain perception, and for the normal and abnormal behavior of any person, this serotonin plays a very important role. Okay, so how are these obtained? They are obtained by the decarboxylation of amino acids. Okay. Now, see, this is glutamate. Glutamate, upon decarboxylation, what you get is GABA. GABA is gamma aminobutyric acid. I repeat, decarboxylation of glutamate in presence of glutamate decarboxylase produces gamma aminobutyric acid. See, this is a neurotransmitter. GABA is a very important neurotransmitter, and this is an inhibitor of nerve signal, and this effect results in reduction of anxiety and induces sleep. So as I told you, the importance of decarboxylation of amino acid is whatever the product they are produced, that is the amines that are obtained through the decarboxylation of amino acids, are important neurotransmitters. Okay, so you should know to write the example for decarboxylation. So write glutamate. Upon decarboxylation, you get GABA, gamma aminobutyric acid. So how do you name it? See, this is a COO minus or an acid group. The carbon atom very next to this is called alpha carbon, alpha, beta, gamma. I repeat, alpha, beta, gamma. At the gamma position, you have an amino group. So this is gamma aminobutyric acid, which is an important neurotransmitter. Now coming to the next process, deamination. What is deamination? Removal of ammonia group is known as deamination. Can take place in two different ways. One is oxidative deamination. So you have an amino acid. In presence of groups like NAD+, this NAD+ which undergoes reduction to NADH, and this gets oxidized. That is, this converted into ammonium ion, and what you get is a keto acid. What you get is a keto acid. They are either one or the other intermediates of Krebs cycle. So the ultimate purpose is to remove this ammonia group in the form of ammonium ion with the help of NAD+. Okay. So glutamate, upon decarboxylation, not decarboxylation, deamination, you are going to get alpha ketoglutarate. Similarly, a non-oxidative deamination, amino acid undergo non-oxidative deamination to form a keto acid. So L-serine, upon non-oxidative deamination, you are going to end up with pyruvate. So which can again enter into Krebs cycle. So what we have learned from this session is, see, these are the examples